So hello and welcome everyone to another one of our master classes. My name is Felipe Merino and I am responsible here at Cube Biotech for our electron microscopy work and services. So for those of you that are new to our masterclass series, these are webinars that we are organizing on a monthly basis. And the idea is to give a platform for researchers from around the globe, right, to come with interesting stories around biotech or biochemistry. So most of our interests go around the membrane proteins, but of course it doesn't have to be that, right? So we have this kind of a umbrella interest in all things biotech. So we have had not only things about membrane proteins, but you also see structural biology, biochemistry, or proteomics, like the case today, right? So if you're interested, if you think that you have an interesting story to tell, and then you would like to use our platform, just drop us a line, and then we can see how we can accommodate your story into our series. So the webinar is going to be organized like this, right? So we are going to listen to the presentation of the speaker today. After that, we're going to have a live Q&A. And the idea with the Q&A is that if you look on the left, in the bottom right of your screens, there is a chat part. Then you can type your questions, right? And below there is a question mark and a sign. So you can basically mark your questions as questions for the Q&A there. So we know exactly that these are the questions that we're going to be asking our speaker today. And then after that, we are going to just ask you for 2 minutes of your time. So if you can answer some questions for us, so we can improve a little bit our webinars more and more. So today we are being joined by Jonas Bossart. He's a PhD student at EMPA and ETH Zurich. He is interested in basically quantitative proteomics of human immune cells, right, from a biomedical perspective. And right now he's mostly working on the phenotypic landscape of macrophages and the response to iron-carbohydrates. So yeah, without further ado, I leave you with today's webinar. So enjoy the presentation.
First of all, I would like to thank the organizers of this masterclass series for having me here today. My name is Jonas Bossart. I'm a computational biology PhD student at the Swiss Federal Laboratories for Materials Science and Technology in St. Gallen, Switzerland, where I am part of the Multiomics for Healthcare Materials group. I am happy to take this opportunity today to present our most recent research focused on the characterization of macrophage phenotypic states and their cellular response to nanomedicines. So let's begin by briefly discussing the importance of macrophages. Macrophages are considered professional phagocytes with diverse roles ranging from destroying bacteria and producing inflammatory cytokines to wound healing. In vitro, one can experimentally induce different polarization states, and on one end of the spectra, there are the pro-inflammatory M1 macrophages, and on the other, the immunosuppressive M2 macrophages. But it is important to note that this M1 M2 binary system is simplified and does not reflect the full biologically relevant spectrum, which is far more complex. For example, the M2 macrophages can further be divided in M2A, M2C, and even more subcategories. In the tumor microenvironment, macrophages can comprise up to 50% of the leukocytes that infiltrate the area. They secrete mediators that may enhance tumor survival, promote angiogenesis, and facilitate cancer invasion. In this regard, the relative fractions of these states are influential in cancer progression. Additionally, their ability to process signals from various cell types contributes to this wide range of molecular phenotypes, showcasing their remarkable plasticity. Recently, the concept of therapeutic reprogramming of macrophages has been gaining more and more attention. But while M1 macrophages are well described, we still face significant gaps in our understanding of the M2 macrophages, which is crucial for advancing cancer treatment strategies. In addition to their roles in cancer biology, macrophages are also important for the uptake of drugs. In this regard, the Kupffer cells, which are the tissue-resident macrophages, of the liver are believed to be instrumental in the uptake and biodegradation of iron-carbohydrate complexes. These iron-carbohydrate complexes are nanomedicines, which are commonly prescribed for the treatment of iron deficiency. They are composed of a polynuclear iron-3 oxyhydroxide core, which are complexed with carbohydrates. There are different formulations and generic products on the market with different carbohydrate compositions, such as iron sucrose, ferric carboxymaltose, and iron isomaltoside. So far, the precise uptake mechanism, metabolism, and the overall impact of these nanoparticles on the molecular landscape have not been fully understood. So we conducted 2 proteomic experiments to investigate both aspects discussed, and both of them are based on primary human cells. Initially, we isolated PBMCs, the peripheral blood mononuclear cells, from healthy donors. These PBMCs were then selected for monocytes, differentiated into macrophages, and polarized to M1, M2A, and M2C phenotypes using LPS and interferon gamma, interleukin 4 and 13, or interleukin 10. Our first experiment was focused on measuring global proteomics and phosphoproteomics across these 3 polarization states to characterize altered protein expressions and signaling pathways. In a subsequent experiment, we treated M2A macrophages, hereafter referred to as M2 for simplicity, with clinical relevant doses of iron sucrose for 45 minutes, 6 hours, and 24 hours. Here, the M2 macrophages were selected as experimental model due to their higher uptake rate compared to the M1 phenotype. The 3 time points were chosen to delineate the temporal dynamics of molecular alterations induced by our main compound of interest. Additionally, we also measured and compared the 6-hour time point following treatment with ferric carboxymaltose and iron isomaltoside to assess cross-sectional differences with these 2 newer drugs. The Functional Genomics Center in Zurich measured the global phosphoproteome and proteome for us. And in short, the workflow starts with protein isolation and trypsin digestion. And for phosphoproteomic, we also need a phosphopeptide enrichment step where we made use of magnetic beads, for example, the iron NTA MAC beads from Cube Biotech. The analysis proceeded with a standard data-dependent and label-free approach, employing high-performance liquid chromatography to segregate peptides by their physicochemical properties, which was then followed by tandem mass spectrometry. We conducted the data analysis in-house to evaluate differential expression of proteins, the overrepresentation of biological pathways, and changes in the kinase activities. I'd like to start the results section with the macrophage project entitled Delineation of Signaling Routes that Underlie Differences in Macrophage Phenotypic States. The manuscript is available as preprint under the following DOI, which can also be accessed directly via the QR code provided. This work describes the first global characterization of signaling events in primary human M1, M2A, and M2C macrophages using mass spectrometry-based proteomics and phosphoproteomics. For the sake of time, I will restrict the confirmation of the polarization states to the M1 on the left in blue and M2A phenotypes on the right-hand side in green. The macrophage populations showed morphological differences, and the applied protocols resulted in phenotypically heterogeneous populations, as can be seen in these 2 bright field images. We then assessed the expression of widely used surface markers by flow cytometry, and we saw that M1 cells had the highest fraction of CD86 positive cells with 85%, while M2A cells had the highest fraction of cells positive for the CD206 marker with 56%. The upper middle graph shows a volcano plot representing the results of the differential expression analysis between M1 and M2A macrophages. On the x-axis, we see the log 2-fold change, and on the y-axis, the minus log 10 FDR value. Here, each dot stands for a quantified protein, while colored ones pass our significance thresholds, with green ones being upregulated in the M2A and blue ones in the M1 macrophages. In total, we found 675 significantly differentially expressed proteins, including the previously by flow cytometry assessed surface markers CD86 and CD206. We found in M1 upregulated proteins to be strongly enriched in interferon and cytokine signaling pathways, while proteins upregulated in the M2A state were enriched in metabolic pathways. My colleague Tiberio Totu analyzed the phosphoproteomics data in detail. In doing so, he made use of the kinase library tool of the Phosphocyte Plus platform to assess if sequence motifs surrounding significantly upregulated phosphocytes indicate enhanced activity of specific upstream kinases. With this, he found, for example, the pro-inflammatory JNK123 and P38B kinases as major M1 phosphoproteome regulators. Using phosphoproteomic measurements, we can also highlight the phosphorylation of specific kinase activation sites that were previously validated in other studies. In the example of PKCα kinase, also known as protein kinase C alpha type, the threonine activation residue at the position 497 was upregulated in both M2 states. And this kinase is known to play a role in anti-inflammatory processes. Combining knowledge from different resources then allows us to construct a signaling network of kinases as measured in our study, here on the right-hand side, for example, for the M2A phenotype. The kinases phosphorylated in the activation loop are colored in light blue, while their first-degree neighbors are shown in dark blue. The network presented here has the advantage that it is constructed at the level of the phosphorylated residues, with both the kinase and substrates being upregulated. We use kinase substrates knowledge from 5 databases to connect the kinases with solid lines, as well as the NetForest prediction tool to complement the missing knowledge with dotted lines. Here we see PKCα as important regulator, and also PAK2 is worth being highlighted, as it was previously reported to regulate development of myeloid-derived suppressor cells in mice. And with this, we contribute to the characterization of macrophage phenotypic landscapes, which could be helpful in the future for defining interventions to therapeutically reprogram macrophages. We then use the generated proteome signatures to classify macrophages in patient tumor samples, which were characterized by single-cell transcriptomics. Here we, for example, see the UMAP dimensionality reduction of approximately 80,000 malignant and non-malignant cells from a published data set by Gonzalez et al. Consisting of 15 brain metastases derived from different primary tumors. We first excluded cells characterized by high expression of tumor markers, annotated the remaining stromal and immune cells, and subsequently isolated macrophages from the myeloid cluster. Top 100 proteins characterized by significant and high fold changes in our proteomics differential expression analysis were then used to estimate gene set activities of each cell. And based on these, we separated M1 and M2-like macrophages. The in M1-like macrophages upregulated genes were, and without considering the ones present in the signatures, enriched in TNF-alpha and interferon signaling pathways, which show the ability to separate pro-inflammatory macrophage populations in a clinically relevant context. Here, the M2-like macrophages, on the other hand, were enriched in the mTORC1 pathway, but their signal is much weaker. And based on this example of the pro-inflammatory macrophages, we think that unbiased proteomic signatures could be a powerful option to characterize macrophages found also in the tumor immune microenvironment. However, it still has to be investigated if including more individuals would increase the power of the signature sets, and if different combinations of stimuli concentrations and exposure times would strengthen the molecular changes. In the second project, entitled Uncovering the Dynamics of Molecular Responses Induced by Iron-carbohydrate complexes in human macrophages using quantitative proteomics and phosphoproteomics, we tackled the question how macrophages take up and biodegrade the widely used iron-carbohydrate complexes. Here we see the interpretation of a generative AI model when we ask it for pictures of iron sucrose, ferric carboxymaltose, and iron isomaltoside, which of course do not represent reality, but it becomes clear that these three formulations have different attributes. So let's start with the temporal dynamics of our main compound of interest, which is iron sucrose, which by the way is the first approved iron carbohydrate complex for patient treatment in 1949. In the following, we will see schemes of affected proteins and phosphopeptides with log two-fold changes in 3 cells besides each gene symbol. The order of them represent the 3 time points: 45 minutes, 6 hours, and 24 hours. White cells are not measured at a specific time point. Gray ones stand for non-significant changes, and green and as well as red cells represent significantly up or down-regulated entries. At the bottom, iron sucrose is shown as intact particle, and we see the representation of iron 2+ and 3+ ions. Our data shows that iron is getting stored in the form of ferritin. In this regard, we found the upregulation of both ferritin chains, the heavy and the light one, to be upregulated after iron sucrose treatment for 6 hours and 24 hours, which is not surprising, but so far has not been shown simultaneously. When it comes to uptake, we found indications that the transferrin receptor 1 is playing a role in the uptake of dissociated iron ions. We not only found an upregulation of STEAP3 residues, which is an endosomal ferrireductase, which is critical for the transport of transferrin-derived iron via DMT1 into the cytoplasma, but also 2 upregulated residues of EPS15L1 and FCHO2, which are required for the cladribine-mediated endocytosis of the transferrin receptor and the downregulation of a RAB11-FIP1 phosphopeptide, which might impair the recycling of the receptor. So together, these changes indicate that the cells are dealing with iron internalized via this route and that they reduce the presence of the receptor on the cell surface. However, this does not mean that the transferrin receptor has to be the main or exclusive uptake mechanism. And indeed, we found also indications that complete nanoparticles are taken up. So the iron composed with the carbohydrates. This includes differences in immunoglobulin, carbohydrate, and LDL receptors. And these receptors either bind sugars or molecules potentially found on the protein corona formed after the nanoparticles are exposed to zero molecules. 2 of these receptors, MRC1 and CD209, but also a third one, CD163, which is involved in the metabolism of heme, are widely used markers for the M2 polarization state, and we found them to be downregulated. Together with the reduced activity of AKT1, believed to be required for M2 polarization, and increased GSK3 activity, which in turn is linked to an M2 suppression, these results demonstrate a loss of an anti-inflammatory M2 polarization. Furthermore, multiple alterations collectively hint towards mechanisms to prevent an overload of labile iron and ferroptosis. As already discussed, the cells store iron in the form of ferritin, which automatically lowers the iron pool. ATM, an atypical kinase essential for ferroptosis, was found to be downregulated after 45 minutes. Its inhibition leads to increased expression of iron regulators, which are involved in the storage of iron. Similarly, a lower CD163 expression might lower the heme uptake, while an increased ABCC5 expression induces its export and heme oxygenase 1, its biodegradation. SLC39A7 with downregulated residues after 24 hours of treatment is a zinc transporter whose inhibition is also known to protect cells against ferroptosis. With regards to the cross-sectional comparison of the 6-hour time point with the different formulations, our data show that the carbohydrate ligand influences the uptake rate and also the loss of the M2 polarization. Here on the left side, we see a Venn diagram with the number of differentially expressed proteins after 6 hours of treatment with the different formulations. These numbers inversely correlate with the serum half-life reported in the literature. We also found the highest upregulation of ferritin chains with iron sucrose treated cells, followed by ferric carboxymaltose, and for iron isomaltoside, we only found the upregulated phosphorylation of the heavy chain. Furthermore, the loss of the M2 polarization became most evident with iron sucrose, including downregulation of 3 surface markers and an M2 inducing kinase, as well as the upregulation of suppressive kinases. With ferric carboxymaltose and iron isomaltosines, these signs are not clear. We hope and think that the gained insights could serve as basis for linking the physicochemical properties of iron carbohydrates with their clinical outcome. And with that, I'd like to thank all my contributing colleagues, collaborators, and supervisors. My PhD supervised by Professor Andrea Limonti and Dr. Maria Bullian. The presented studies have been led by Dr. Maria Bullian, Dr. Vanessa Ayala, and Professor Peter Wick. And of course, I'd be happy to take any questions.
Yes, so thank you very much, Jonas, for that very nice presentation. Could we, let me see, activate Jonas's webcam? Ah, there you are.
Hello.
So thank you very much for the talk. I would like to maybe start with a few questions myself before we go into actually the Q&A. So, well, do you have any plans to validate some of the proteomics hits that you got, especially in the last in the last story that you mentioned? Is there a way of doing that?
I'm sure. I mean, that's very important, but I won't perform the experiments myself because we are a team with broad expertise in different fields and nanospecialists. Of course, it's important now to validate the receptors because we are especially interested in the uptake. But it's a really complex system, and
Right.
we think that we are facing a situation where we have multiple iron species at the same time and multiple uptake mechanisms to be involved, and this makes it really a challenge. But yeah, we are on it, definitely.
Right, and basically related to that, now that you're providing basically an overall understanding of how the uptake of these carbohydrate iron particles are incorporated and stored, do you think this could help to design better kind of iron delivery systems, or do you see a way to go forward with that?
At least that's what we are hoping for, of course. I think if we understand better how the complexes, the carbohydrates influence the uptake and also the other molecular alterations like a loss of M2, basically we would gain a rationality to design better drugs in the future, of course, but that's far, far away.
Right.
That would be the idea, definitely.
Probably the system is super complex and this would take some
Indeed, yeah.
serious effort. Okay, then let's Q&A is here, right? Okay. So Henning Beckhoff asks, thank you very much for the presentation. As far as I understood, you performed the proteomic analysis with blood-derived monocytes differentiated in vitro. Do you know to which extent the results holds true for Kupffer cells in the liver?
Really good question, though. No, I can't really say. I mean, it all depends on the health status of the patient, I guess. We selected M2A macrophages to perform these experiments just because we measured by ICP-OAS that they take up the nanoparticles more efficiently. And with that, we were hoping that we get the most meaningful results because the cells already took up the nanoparticles. If we take others, it takes a longer period of time and the changes might be difficult to interpret, sorry. Yeah.
So Sarah Ames asks, could you please repeat the effect you hope to trigger by analyzing the macrophages in tumors?
The effect. I guess you're referring to the annotation of the macrophages. If that's true, so the idea would be that we use our proteomic markers, the signatures we measured, and we want to separate different clusters or different groups of macrophages in the tumor immune microenvironment to better understand how they relate to in vitro generated signatures. Because it's really, really difficult at the moment for physicians to really guess which macrophages acting in which way, because as you know, in the tumor immune microenvironment, the macrophages follow a broad spectrum, and it would be helpful to at least partially classify them in different groups.
So Martin Johnson asks, what is the difference between the M1, M2A, and M2C cells you mentioned?
Okay, so these three cell states are in vitro generated, so they're not really physiological, but we can generate them in the lab. And with M1, we have a pro-inflammatory subtype, and with M2, on the other hand, we have the anti-inflammatory functions. And M2A and M2C, they differ slightly, and they have different roles in angiogenesis and wound healing, for example.
Cool. So Charles Pano asks, thank you very much for the presentation. How relevant are the results for obtaining a phosphor profile of the different tumor microenvironments?
So maybe I was not really clear. I mean, thanks for the question, but we did not measure phosphoproteomics of the tumor microenvironment. This would be a big challenge because I guess the fraction of macrophages is still low in a tumor microenvironment. Right. And for phosphoproteomics, we require relatively high protein input, at least with the technique we used. So we used at least 100 micrograms of tissue, which would be difficult to get, actually. And also, we would need to sort the populations, maybe by fax, but we would then bias the analysis, of course, by specific surface markers.
Okay, so maybe we have time for one last question. This is from Carmen Castillo. She says, I'm planning a project involving phosphopeptide enrichment as well. Would you say the nickel MTA MAC beads are work intensive?
Unfortunately, I can't really answer this question because so we work together with the Functional Genomics Center in Zurich, and they actually performed the measurement for us and also the enrichment of the phosphopeptides. I guess Felipe can better answer that question than myself.
Yes, I would say they're wonderful.
well,
yeah. Okay, then if there aren't any other questions, then I would like to thank you again for your talk and for your time. This was very interesting as well.
Thanks a lot.